The thing about translocation is that you spend so much time studying transpiration, and then there's just 2 little lessons tagged on for the end for translocation, and it's like, oh, nearly the end of plant transport. And for too many people, the risk is too high to just sprint through translocation because it's just tagged on and miss it and not get that depth of detail that you need. And yet, translocation is a topic that links so nicely into so many other earlier topics that it's one of those beautiful topics for explain questions. And when it comes to an explain question, it's really all about that detail. So if you rush through translocation, then you don't have the detail. Now you've got a problem. So this lesson is going to be looking at that detail of translocation, highlighting it to make sure that it has a level of importance in your mind, and that you remember it going forward. In order to kick off this, we need to actually have a look just briefly at structure when we come to the phloem. Okay, remember in your phloem, you've now got living tissue. Xylem is your dead tissue, phloem is living. And if we take a cross section, we might have something like this with a cell wall because it's a plant cell. We don't use rulers, but we always draw plant cells with a double line for the wall. Then next to it, you would find a smaller cell, also with double line for the cell wall. So who are these two? This one here is the companion cell. And this one here is the sieve element. And these two work so closely together that it's crazy. The sieve element doesn't function without the companion cell. So if we have a look in a little bit more detail to look at why this happens, but I'm not going to draw that one. I'm going to open that one separately. So here we are looking at a more detailed diagram. Now you can understand why I'm not sitting and drawing the whole thing in this lesson. Gives you a bit more detail now of a transverse section of a sieve element and its companion cell. Remember, these are really elongated cells. They're also transport cells. Big thing is, if you look at these two, even though we've got 2 cells, we only got 1 nucleus. Here in the companion cell is a nucleus. Vacuole, only in the nucleus. Rough ER, only in the companion cell. Sorry, that vacuole is also in the companion cell, not in the nucleus. Obviously, it's been one of those mornings. Right, so we have rough ER, we have vacuole, we have nucleus, we have ribosomes, all going on in the companion cell only. In our sieve element, we've got mitochondria, we've got smooth endoplasmic reticulum, and we have cytoplasm. But see how it's sort of roughed in? The cytoplasm is only around the edges. Cytoplasm is around the edges. In the inside here, we have the phloem sap. The phloem sap is very simply the liquid that's inside the sieve element. So we're going to talk a little bit more about that in a moment. For now, that is the reason why we only have that thin layer of cytoplasm lining the inside of the wall of our sieve element, because we have phloem sap moving in between. Okay, we've also had a question come through of how is the cytoplasm separate from the phloem sap? So that's how is jelly separate from custard? Okay, remember that your cytoplasm and your phloem sap have different viscosities. So they're not going to mix directly. Makes sense? Other important things to note is you've got lots of mitochondria. Okay, this is not your standard amount of mitochondria in a plant cell, and you've got lots of plasmodesmata. Okay, so think back to organelles and what's the functions of your mitochondria. Production of ATP, aerobic respiration. What's the function of plasmodesmata? It is for unblocked, no membrane involved, transport between cells, which is faster. Bear that in mind. Okay, then between one sieve element and the next, as we go up and down the plant, we have sieve pores. So if we go back to our other diagram, here it is. So the sieve pores and the sieve plate would be across the cell. So all you would see if we were looking at a sieve plate is holes. And that's then the sieve pores. And these are really important because if the plant stem is damaged in any way, we can block these pores and therefore we can stop the loss of that phloem sap. Okay, phloem sap has got a lot of investment by the plant. All these products of photosynthesis, all the things that the plant has put energy into making. So we don't want to be losing them. Phloem sap is very important to the plant. Okay, so you've got a good idea now of an overall of the structure. Yes, you do need to get that detail really sort of into your head. It is, you need to know this, okay? But the sort of take-home message here is that companion cell and the sieve element, they work together as one. You can't do this whole job of translocation without having both of them. So let us now take a moment to look at phloem sap a little bit more and translocation itself. So what is phloem sap? Then we talk about it. Phloem sap is water plus assimilates. So we have assimilates, which is things that the plant has made, okay? So these are your sucrose, amino acids, even ATP. We also have callose. Okay? Callose is what actually blocks those sieve pores if there's a problem and the phloem is broken. So all these things are making up the phloem sap. Okay? All of them are in the water. So we have a very important linkage here, okay, between our assimilates and our water. Okay, so all of these things make up phloem sap, but often we sort of simplify the world and we talk predominantly about sucrose. Okay, so we talk about translocation being the movement of assimilates, but when we get into the details just now, we're going to focus on sucrose. Okay, so how does phloem sap move? How does this translocation happen? So phloem sap moves by mass flow. Okay, so phloem sap is moving by mass flow, but it is not the same as mass flow in xylem for transpiration. Okay, so transpiration is loss of water, and then water comes in and it just moves by mass flow. Phloem sap moving by mass flow is actually an active process. So it's very different to the mass flow that is created in water by that loss of, created in xylem by that loss of water. Okay, in this case, we actually have to use energy to create a pressure difference between the source and the sink. Okay, so let's have a little bit more detail on how that actually happens. Okay, so we have at the source and we have at the sink. At the source, we have sucrose is loaded. So we're already in that simplification. Okay, these are your leaves, your storage organs. Okay, anywhere where sucrose is produced or released is somewhere where it can become a source. At the sink, this is where sucrose is removed. So this is anywhere where sucrose would be needed. So we're talking about anywhere with growth, anywhere we've got extra energy use. So we can be removing in the roots, in the buds, in the flowers, in the fruits, in the storage organs, even in growing leaves. Because if you can imagine it, when a leaf is really still very small, it might even have a higher need for sucrose than it is able to produce by photosynthesis. When that balance goes out, then that leaf becomes a sink because it's drawing resources from other parts of the plant. Okay, think about leaves when they very, very first start growing, where you've got buds rather than open leaves. So you can understand how sources and sinks can actually even change between different stages of the life cycle of the plant. Okay, so at the source, we're loading sucrose. At the sink, we're removing sucrose. And sucrose, remember, is a relatively large molecule. It can't just diffuse across the cell membrane. And the active process comes in actually in this loading. Sucrose is actively loaded into the phloem. That then causes the water to follow, and then we have a creation for mass flow. And then we get mass flow from the source to the sink. So it doesn't matter where in the plant that sink is. If the sink changes location, if the source changes location, it doesn't matter. Because the source is always going to be working in conjunction with a sink over a pressure difference. And this is what's really important about translocation. So we're going to go into that in a bit more detail now about how that process actually happens, how the sucrose is loaded, and how that then leads to this difference that causes the mass flow. Okay, so being asked the question that storage organs can be a source or a sink, and yes, that's absolutely right. So if you think about it, a storage organ, if you think also maybe in somewhere like Europe or America where you've got real seasons going on, not necessarily like we have here. Okay, so in the summer, in the spring, we've got lots of growth. Okay, in the summer we keep going with lots of growth, but we're producing more than we need, so we store it. Because then through the autumn and then into the winter, we can actually use nutrients. And when we're ready now for that great big growth spurt in spring, we've got the sucrose and other nutrients, other assimilates, also waiting around to be used. So at certain times of year, that storage organ, we will put things into storage and then we will use them again. So when we're putting them into storage, it acts as a sink. And when we're bringing them out of storage to use, then it becomes a source. Cool. All right, so coming back now to translocation and transpiration, just a little bit more, another highlight. Okay, transpiration is always in one direction, okay, always from the roots to the leaves. It's a passive process, it just happens. Because translocation is an active process, even though it's also mass flow, because it's active, it can go in whichever direction it needs to be. So it can go source to sink as we putting things into storage. It can go, then the storage organ can become the source and we can move in the other direction. And that is the value of having it now as an active process, is it something that's controlled differently. And therefore we can have a different output. Let us have a look then and a bit more detail about how we do this active processing that brings us the mass flow, that starts this whole process off. Okay, so the big deal and where everything happens is in the loading of the sucrose. Okay, so how is this an active process? H+, your hydrogen ion, okay, he is actively transported out of the cell, the companion cell. So remember, if you want to know it, this is a proton pump that does this one. Because we always have a specialized protein to do any specialized transport job. So the specialized protein that is actively transporting hydrogen ions, protons, out of the companion cell is called a proton pump. Bear in mind also that this out of the cell is just across the cell membrane. So if you think about plant cells, you've got a membrane, then a wall, then maybe a space, then a wall, then a membrane. So there is actually space between membranes of neighboring plant cells. And that's where these hydrogen ions go. They go into that space between. Okay, but active transport is against the concentration gradient, which means that those hydrogen ions, they want to come straight back in again. Okay, but they're ions. They can't just go across the membrane, because our membrane is a phospholipid bilayer and it has a hydrophobic core, which doesn't like ions. You see how we're tying things together? So hydrogen ions have to get back into the cell via another protein, and this one is called a co-transporter protein. Nice to remember that, because if you co-transport something, you're going to transport it together. So hydrogen ions return via co-transporter protein. Okay, so what comes with that hydrogen ion? A sucrose. So the hydrogen ion who wants to get back in can only get back in when he's with a sucrose. So now you're building up all this sucrose in your companion cell. Now sucrose is a great big molecule. It's definitely not going to be able to diffuse across the membrane, but we have this massive influx of sucrose. We have this high concentration of sucrose, so sucrose is going to want to diffuse somewhere into the plasmodesmata. Okay, remember, plasmodesmata has no membrane across it, so therefore the sucrose can just diffuse straight across the companion cell into the sieve element. So it diffuses across the companion cell, through plasmodesmata, into the sieve element. And just stop and pause here and think about transport across cell membranes. We've got active transport here, we've got facilitated diffusion, and we've got diffusion. So, wonderful question if you're going to be asking about transport across cell membranes. Okay, so now we are in the sieve element. Now we've got a whole lot of sucrose in the sieve element. So our Phloem Sap has increased in concentration. We have dropped its water potential. We couldn't just stop at 4 modes of transport across the cell membrane. We've got to add another one. Okay, as soon as we're talking about water potential, there's only one method we can be talking about, and that is osmosis. So as soon as that concentration goes up, water potential goes down, then water just wants to enter that sieve element by osmosis. And what happens when you put a whole lot of water into a relatively small space? Your pressure increases. So all because hydrogen ions were actively transported out of the cell, we end up now with an increase in pressure. Now, if we increase pressure somewhere, something's got to give. So remember that your phloem is one long tube, cells joined over and over and over again. So we can move. So our next step is movement. So we got a high pressure place. Where are we going to move? We're going to move somewhere with low pressure. So Phloem Sap moves from high pressure to low pressure by mass flow. So we actively create that pressure difference, and then the movement happens. So then we get to the sink, and now we have to do the unloading. Here we don't go into as much detail. What's rather funky is, last time I checked, they didn't know the detail for this one. There's a whole area of research open for you guys to go and figure out what's going on. Okay, so essentially, sucrose is removed. Okay, that's the process where we don't have the details yet. So put it on your to-do list. Okay, obviously, as soon as we remove that sucrose, our water potential is going to increase because we're dropping the concentration of that phloem sap. So the water is also going to leave. And when the water leaves, we have the pressure decreases. Okay, so now you could imagine, okay, well, the sucrose is going out, but then we just have a buildup of sucrose in the cells. But remember, we don't want that sucrose hanging around. It's going to get turned into other things, it's going to get used by other things, and that's going to maintain that pull of sucrose out for as long as the cell needs to pull, or the tissue needs to pull sucrose out of the phloem. It's going to be using it or converting it. Sucrose is our special disaccharide, broken down into glucose and fructose. And then we're talking about things like respiration, we're talking about cellulose, we're talking about starch. And obviously we've got an issue here, we've got fructose, we've got alpha and beta glucose. So we know there's a lot more detail going on. But the level at which we're studying it, we're just looking at the idea that these things are used in other processes once they're unloaded at the sink, so that we can maintain this pressure difference. At such a time when the sink no longer needs products of our phloem, then it will stop removing them. So that pressure then will normalize. So somewhere else is going to then have a lower pressure spot, and the phloem sap will go in that direction, depending on where it's needed. So because we have this actively controlled entry and exit, then it means that we can change those entry and exit points depending on what is happening in the tissues surrounding the vascular bundles at the time. So it's a much more finely tuned, much more controlled system, in which is what makes it so much open to our detailed explanation questions, because there is all this going on about the control and different types of transport